Hello friends, in the previous video we discussed about elongation step of eukaryotic DNA replication. You can refer my video, the link is given in the description box. In this video we are going to discuss about termination step of eukaryotic DNA replication. So what we know that after elongation, termination takes place and it gives two separate copies of DNA. And as the two separate copies of DNA are obtained, this marks the completion of DNA replication or the end of the DNA replication. In this process or in termination, DNA synthesis is completed, the replication machinery is disassembled and daughter molecules are resolved. Now the termination sites, it is determined by position of replication initiation sites. We know that there are multiple origin of replication and hence multiple replication bubbles are formed, and the site where the adjacent replication bubble converges or meets or the adjacent replication fork meets is the termination site. But in case of the prokaryotes, there is specific locus known as the ter site where the termination takes place or the two replication fork meet, and this is diametrically opposite to the origin of replication. As in prokaryotes, DNA molecule is circular, but in case of eukaryotes it is linear. So this is the linear DNA molecule. There are multiple origin of replication, and as the two strands separate at the origin of replication, then the multiple replication bubbles are formed. Strands in red color are the parental strand or the template strand, and those in blue color are daughter strands. Each replication bubble has two replication fork which move away from origin in opposite direction to each other. Hence, it is bidirectional. When the adjacent replication bubble, or in other words, when the adjacent replication fork meets or converges, then the termination of DNA replication takes place. Now let us understand this with the help of diagram. We have taken two origin of replication, and when the strands separate, two replication bubbles are formed. Each replication bubble has two replication forks. As the helicase proceeds, two strands separate and hence the replication fork also moves or proceeds, and both the replication fork of the replication bubble move away from origin in opposite direction. So replication fork of this replication bubble, one will move in this direction and one will move in this direction. Similarly, if we talk about this replication bubble, the replication fork will move in this direction and this replication fork will move in this direction, that is opposite to each other. When the replication fork of adjacent replication bubble meets or when the adjacent replication bubble converges, then the termination of DNA replication takes place. So as the helicase move and the DNA strand separates, then the leading strand of replication bubble 1 reaches the lagging strand of the replication bubble 2. And in case of the lagging strand, as the lagging strand is synthesized in the form of the Okazaki fragment, so this Okazaki fragment will reach the 5' end of the previous Okazaki fragment. This is the 5' end and this is the 3' end. In this way, the process continues. Similar will be the case, the leading strand of this replication bubble reaches the lagging strand of the first replication bubble. So what is actually happening? The replication fork of this replication bubble is moving in this direction, and for this replication bubble it is moving in this direction. So this stretch of the DNA between the adjacent replication bubble 1 and 2 is also replicated, and when the DNA polymerase reaches a section of the DNA template that has already been replicated in case of this replication bubble and also in case of this replication bubble, then the DNA polymerase halts. That means this entire section of the template is replicated, and thus the termination of the replication takes place. Similar will be the case for the other adjacent replication bubble, as the eukaryotic linear DNA has multiple origin of replication and hence multiple replication bubbles are formed. Now let us discuss the events that takes place during termination. As discussed earlier, adjacent DNA replication forks or replication bubbles converge or move towards each other during termination. This results in unwinding of stretch of DNA between two adjacent forks. So, as the two adjacent replication fork converge or move towards each other, the stretch of the DNA which is present between the two adjacent forks unwind. CMG complexes along with associated proteins approaches each other. So, CMG complex, which is functional replicative helicase, results in the unwinding of the stretch of the DNA. Hence, as the replication fork proceeds, CMG complexes, which is associated with other proteins, approaches each other, or these proceed towards each other. Then these CMG complex bypass each other. This is because synthesis of lagging strand is slower compared to leading strand, and hence certain stretch of the DNA is unreplicated, as a result of which CMG complex along with other proteins including the DNA polymerase bypass each other in order to replicate the unreplicated stretch of the DNA. Then these are associated onto the double-stranded DNA. This is followed by the processing of Okazaki fragments, means removal of the RNA primer and replacement it with the stretch of the DNA. This we will discuss in detail in separate video, removal and replacement of RNA primer. This is followed by recruitment of SCF E3 ubiquitin ligase complex. This SCF catalyzes the ubiquitination of MCM227 on the 7th protein. So we know that MCM227 is hexamer. It consists of 6 monomeric subunits numbered from 2 to 7. So this SCF catalyzes the ubiquitination of 7th protein of MCM227 or MCM hexamer. So normally what happens? Ubiquitinated protein is degraded by proteasome, which is known as proteasomal degradation. But here this ubiquitinated MCM227 is recognized by P97, also known as valucin containing protein, in short VCP. This is segregase protein or segregase enzyme, which causes the disassembly of CMG complex along with its cofactors and other proteins. Hence, these are removed or segregated from the DNA strand. Then any catenanes in the daughter DNA strands are removed by topoisomerase 2. This we have already discussed in topoisomerase video. You can refer my video, the link is given in the description box. Therefore, two newly synthesized strands are segregated, hence 2 copies of DNA are obtained. Now let us understand this with the help of the following diagram. Strands in red color are parental strand and those in blue color are the newly synthesized strand. Stretches that is shown by orange color are RNA primer. This is the CMG complex. During the termination, what happens? The two replication fork move towards each other. Arrow represents the direction of the movement of the replication fork. That is, these converge each other. As a result of which CMG complex also along with other proteins converge or move towards each other. CMG complex, which is functional replicative helicase, causes the unwinding of the double-stranded DNA. It is also associated with other proteins including the DNA polymerase. Hence, new strand is synthesized. These encounter each other. Then these bypass. Here, for convenience, we are just representing the MCM227, but it is actually CMG complex. So these bypass each other. This is because leading strand is synthesized faster and lagging strand is synthesized slower. Hence, these move away from each other in order to synthesize the stretch of the template that is unreplicated. Once the synthesis of new strand is complete, then the processing of Okazaki fragment takes place. That is, RNA primer is removed and it is replaced with stretch of DNA. This we will discuss in detail in separate video on primer removal. Then recruitment of SCF-E3 ubiquitin ligase complex takes place. This is SCF. This causes the ubiquitination of MCM2 to 7 at the 7th protein. Now this ubiquitinated MCM2 to 7 is recognized by P97, valusin containing protein, which is the segregase enzyme or segregase protein, and this causes the segregation or disassembly of CMG complex along with its other cofactors and proteins. This is followed by the decatenation of the newly synthesized strand. That is, if any catenins are present between the newly synthesized strand, then it is removed by topoisomerase 2 and hence the two newly synthesized strands are separated. And this gives two double-stranded DNA molecules. This is all for today's video. In the next video, we will discuss about primer removal and its replacement by DNA. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.